And then we'll get microphone.
Great. Hello, everyone. I'm very excited to introduce Dr. Yasir Ahmed-Braimah, who's a PI at Syracuse University and a member of the Center for Reproductive Evolution. He was recently awarded the Early Stage Investigator MIRA grant from the NIGMS. Congratulations. He got his bachelor's and master's at the University of Iowa, where he studied philosophy and biology. That's where he became interested in speciation and reproductive isolation and incompatibilities. He joined Bryant McAllister's lab, where he studied positive assortative fertilization as a result of allopatry between Drosophila americana and Drosophila novomexicana. And then he did his PhD at University of Rochester, where he worked with Alan Orr on the genetics of species differences in Drosophila virilis group. And then he did his postdoc with Andrew Clark at Cornell University. And now he's going to be telling us all about the genomic basis and molecular mechanisms of reproductive incompatibilities at Syracuse University.
Thank you. Oh, that's nice. Thank you so much. Super happy to be with you. Let me make sure this is not blocking the slide. You can hear me okay? Okay, excellent. All right, well, thank you so much for the invitation. I'm super happy to be with you guys. What I thought I would do for the seminar is tell you about what we do here in the lab. We have several projects, but I'm going to focus on our speciation projects. And I'm going to actually do something I haven't done before, which is tell you about a very new project that we haven't published on and I haven't really talked about before. And so it's a bit of an experiment. So I'd love any positive feedback or negative feedback too. It's fine, I guess. Right. So I, like most of you, am very interested in what creates biodiversity. And, you know, I think many of us study this at very different scales and different methodologies. I think to me, it's really remarkable that we can actually explain the generation of biodiversity at a reductionist level. And that's kind of where I got my start. I was very much just interested in knowing how do new species form. Now, Rose mentioned that I studied philosophy, and actually in philosophy, there's this sort of conundrum about what is a species. We're not going to talk about that today, obviously. So I'm going to assume that we all agree that species are a gradient of lineage differentiation between different groups. So for me, really, the simple abstraction of how species form, as I think you guys all have this intuition, is that an ancestor splits into two different populations that start to accumulate some genetic differences. And here, this sort of simplistic phylogenetic abstraction shows you that along those diverging lines, we expect that some genetic differences are accumulating. And at some point, those genetic differences lead to those two new species being quote unquote species, right? For us as, say, molecular geneticists, we're interested in actually identifying the molecular mechanisms that cause them to be species. For that reason, we use sort of reproductive isolation as a proxy, right, for what species are. I think it's a pretty good proxy, even though I think you can have species that are not fully reproductively isolated. So it is a bit of a gradient. At least it gives us some framework by which to identify species and study them in the lab. Okay, so I'm guessing everybody knows what reproductive isolation is, but let me just sort of tell you how we think about it. So if you have a male and a female from two different lineages and they come into contact, many things can happen that could lead to reproductive isolation. So the initial thing is, the reproductive rituals that take place might be incompatible in some way, and that might lead to some sort of isolation. So whether this be behavioral or gametic, we term these prezygotic barriers, right? And if the hybrid that they make, if they make one, suffers any sort of fitness disadvantage, then that would be considered some sort of barrier, right? And so that's where the prezygotic versus postzygotic terms come from. And as some people suggest that these are arbitrary, but I actually don't think so. Prezygotic barriers are really an incompatibility between the phenotypic manifestation of two different genomes in two different individuals, whereas postzygotic barriers are incompatibility between two genomes in the same cell, right? So they're conceptually, I think, very important to keep that in mind. And keeping with that theme, when we think about postzygotic barriers, there's actually a pretty well worked out, very simple model that suggests that postzygotic barriers have to be at least the product of two loci that interact in these sort of negative epistatic interactions. And the way those happen is that throughout the two lineages, the two loci fix two different alleles that have never seen each other in the same genetic background, right? And so if you bring that hybrid, those two different new alleles into the same background, that might result in some sort of defect. Okay, so this is the Dobzhansky-Muller model. I'm sure you guys heard a lot about it. Now, we don't have a similarly elegant and important named model for prezygotic barriers, but I think the intuition is very clear. If we think of reproductive interactions or any sort of reproductive ritual between, sexually reproducing organisms, there is a kind of coevolutionary dynamic between males and females. It's not important what that dynamic is. It could be some people like arms race, some people like genetic conflict, some people like synergistic, interactions. Whatever it is, there is some genetic link between males and females due to this reproductive coevolution, right? And if we imagine that that happens independently in can you see my pointer, by the way? Yes, good. Okay. If those coevolutionary processes are occurring in these two lineages independently, when a male and a female from those two diverging lineages then come together, there might be some incompatibility, right? So this doesn't have a name, but let's just imagine that that's kind of a pretty reasonable intuition. So we study this process in the genus Drosophila. I'm sure many of you have heard of Drosophila before, but I want to kind of remind you that there's lots of Drosophila species out there, over 2,000. Here's a mostly accurate representation of Drosophila species in the phylogeny. This is a tree that a student of mine built using a bunch of genomes. And you might be familiar with Melanogaster, which lives up here. There's a lot of interesting biology across the Drosophila tree, and I won't go into selling Drosophila to you, right? But we work down here in the Drosophila subgenus. So this entire genus is called Drosophila, and then there's a subgenus also called Drosophila. Melanogaster is in Sophophora. And we work with a group in the Repleta virilis radiation. It's called the virilis group. And as you'll see, it's a wonderful system, not just for studying speciation, but for a variety of different genome evolution questions that I won't talk about today. So we know a decent amount about the biogeographic history of the virilis group. And this is going to be kind of important for the thesis of my first topic here, which is that I'm assuming that these species have evolved in allopatry with minimal to no gene flow. And again, I appreciate that speciation can happen in many, many complex ways. We're using the most simple approach, which is assuming no gene flow in the past. And so in this map, you can see kind of what we know about the distribution of those species. Here's the quartet of the virilis group. There's kind of two subgroups within the virilis. This is the virilis subgroup. So there's a virilis group, and then there's a virilis subgroup. It's kind of hard to keep track of. And the group itself originated in northern Eurasia, and subsequently this lineage of Americana and Nova Mexicana, as the names imply, have crossed into North America and have settled either side of the Rocky Mountains and maintained allopatric distributions there. Okay, so as far as we know, gene flow is fairly minimal in the recent past. The namesake of the group, Virilis, also originated in Eurasia. In the last 100 years or so has become a bit more cosmopolitan, if you will. Not as much as Melanogaster, because they're not actually easy to find. I've never been able to collect them anywhere, really. But you see there's massive sampling in Japan. And through movements of wine and sake and beer, these guys are typically where you find them, right? So I've gone to several wineries and breweries. It's very hard work, right? To find these guys. Never found them, and so they're really hard to collect. Okay, so but in general, we think that over the past they've evolved largely in allopatry. There may be some recent gene flow now. Okay, so why are they good for studying reproductive isolation? Well, we collected a bunch of data and also using data from the past, spanning really 60 years. We compiled all that we know about the 4 different broad-scale reproductive barriers that I highlighted before. On this table here, I'm just showing you all the possible pairwise combinations you can do between members of this group. And now I'm going to show you a dot plot where the size of the dot indicates how strong that isolation is, okay? So just by seeing this, you can immediately notice one thing, is that these types of barriers here, these post-mating prezygotic, or otherwise known as gametic isolation barriers, these are very, very rampant, right, in nearly all crosses. They vary in severity from, 30, 40, 50% to 100%, right? So what's interesting about this is, well, why does this evolve so rampantly in the species? You'll see that there's a scattering of other reproductive barriers here that are prominent. We work on all of those with the exception of this one. So, but why does this evolve at such a high rate? That was always the question that interested me. So they become really an ideal system to do this. And I should emphasize that this is non-competitive gametic isolation, right? So in heterospecific crosses between two species, you see this. You don't need competitive context, for example, by including a conspecific cross within the heterospecific cross to see this. Okay, so I'll tell you about the work that we're doing in this area. So the first part I'll talk about is our efforts trying to understand the molecular genetic basis of these incompatibilities. So I'll tell you a few different stories about that. And then I'll also tell you about more recent work that we're doing on this hybrid sterility phenotype that is really elegant kind of trade, because it's the first time, at least that I know of, where a hybrid sterility phenotype is involved, completely involved, results from a Y-X incompatibility rather than an X autosomal incompatibility. You'd think, wait, so the Y chromosome, like, that's all it does. It just does male fertility. How come that's never been, right? I thought the same thing. So it's a really interesting opportunity. Okay, so let me motivate this first part by kind of saying maybe why I think the prezygotic barriers are quite interesting and why it's important to understand how they evolve. I think it's maybe the consensus view among, if not all evolutionary biologists, most of them, that sexual selection has some big role to play in creating phenotypic diversity, and obviously, sexual dimorphism. And one would suspect that if that dynamic is occurring in diverging lineages, that it might result in some sort of behavioral isolation or prezygotic isolation, right? And this has been appreciated for a very long time. I can flash, hundreds of images here of weird organisms with weird ornaments or weird behaviors or very strange horns, or right? And we will immediately say, oh yeah, the males look like that and the females don't, or vice versa, which is rarely. And then we would conclude that that is evolving due to sexual selection, right? I mean, the one example I'll show you is just birds of paradise, for example. If you haven't seen weird stuff like this, you can go to YouTube. There's tons of stuff out there. And we all, I think, very confidently conclude that, yeah, these males have these ridiculous traits because they need them for mating, right? They need them during courtship interactions and so on. And the extent to which those traits result in reproductive isolation is what we really don't appreciate well, or we don't understand, at least at the molecular genetic level. Now, behavior is hard, and of course, working with these birds at the scale that we're interested in is also very hard. And so I've always thought, well, I think if we work on the same kind of problem, but at a different level, we might actually make some progress. I'm not saying we're not going to make progress here. It's just not me. If you appreciate that sexual selection actually takes place even after mating, right? And this is sort of the field that started in the 1970s with the proposition that sperm competition can lead to very divergent reproductive traits, right? And that has a very analogous kind of systematic way of looking at sexual selection compared to, say, behavioral isolation, right? The idea is that you have a valuable resource that is rare, i.e. the ova, right? And a cheap resource that is abundant, i.e. sperm. And then males' reproductive interest is to maximize the distribution of that sperm. And females, their primary interest is to, choose quality males, and so on and so forth, right? This process is both at the behavioral level and at the gametic level, and I hope you can appreciate that that's the case. Of course, now, there are some nuanced differences and so on, but I think at this closest approximation, that's a pretty good, reasonable expectation. So how rampant is post-mating, post-copulatory sexual selection? And I think it's happening in any species where females mate with multiple males and are able to store sperm for prolonged periods of time from multiple males, right? And so this is basically nearly all insects, right? And some birds, and there's some mammals. We obviously don't have long-term sperm storage organs and so on. So whenever those sort of conditions are met, I think you can expect that there's going to be some post-copulatory sexual selection, in which case you might expect that drives differentiation of reproductive genes between lineages. So if you're not convinced, let me just show you a video from my colleague down the hall. Drosophila obviously do this, right? Female Drosophila will mate with one male. They have specialized reproductive organs for storage that I'll show you in a second. And they can store sperm from multiple males. And so that sets the arena for competition between those sperm, and also for females to exert selective forces on that sperm. So this is just a video showing two males that have GFP tagged sperm heads. So you're only seeing the heads. There's green, and it may be harder to see red sperm that are, kind of interacting. And obviously now they're competing for fertilization. This is a bit of a kind of Hollywood look at the scene. It's much less elegant than that, right? This is an opportunistic case where the reproductive organ is sitting like that, and usually they're just anchored near the opening, waiting for an egg to land in the bursa. Okay, so post-copulatory sexual selection is rampant. It can lead to reproductive isolation at the gametic level. That's at least what I think is happening in the Virilis group. The question is now, well, how does that actually happen, and what is the genetic basis of that? So before I show you what we know about why there is reduction in fertilization success between species, let me first kind of make sure that we're all on the same page as far as how reproduction works in flies. You can probably guess how it works. It kind of works the way most of it works, but I just want to kind of introduce some terminology that I'm going to use throughout the talk. So this is the male reproductive tract. There are three main components: the testes, which produce sperm, the accessory glands, which produce seminal fluid proteins and other stuff, and the ejaculatory bulb, which also produces some seminal fluid proteins, but also produces the mating fluid. Okay, so the sperm actually obviously gets mixed with the seminal fluid and material from the ejaculatory bulb. That gets transferred to the female, and here's a female reproductive tract. The main things to remember are that the female has this bursa where the male ejaculate is deposited, and then the sperm can either travel to one of two storage organs. So the spermatheca we think of as the long-term storage organ. These are these two mushroom-shaped things. And then the seminal receptacle, which is a much longer tube, and that length co-evolves with the length of the sperm. Okay, so across the Drosophila tree, you can see that. And these are the two main storage organs. We think the spermatheca are the long-term storage organs, and we think that the seminal receptacle is the fertilizing set storage, right? And I'll show you kind of a model of how that works. Now, females also have glandular structures. So the spermatheca actually does have secretory cells, and also has these paravaria, which are also secrete material into the bursal lumen. And of course, there's ovaries where the eggs come from. That's fairly trivial. Okay, so here's what the phenotype looks like when we look at two members of the Virilis group. I'm looking now at Drosophila virilis and Drosophila americana. We measure the incompatibility by just measuring the hatch rate, which is a perfect proxy for the fertilization rate, and we've demonstrated that in a couple of papers. So obviously within species, the fertilization rate is high. So here's americana fertilization rate, virilis fertilization rate. But between species, there's a dramatic reduction, and that reduction is asymmetric, right? So there's about 25–30% fertilization success when americana is the female, versus when virilis is the female. So it's americana males have a difficult time fertilizing heterospecific females. Whenever I've given a talk in Europe, I always, just so that this sticks to people, I say, just imagine American males are very bad at fertilizing foreign females, and that always sticks. So it's not as funny here. Okay, so the main thing we want to understand is what is causing this reduction, right? Virilous females with virilous males, totally fine. But when it's an Americana ejaculate and sperm cocktail, there's this dramatic reduction. So early on, we were looking into this during my PhD. We didn't have a lot of tools, so we would just dissect out reproductive tract and look to see whether there's sperm there. Sperm obviously are in a massive bundle, so we can't really quantify them very well. But the phenotype is actually super clear, right? Here's a female reproductive tract where it's a virilous female mated to a virilous male. And you can see lots of sperm in the seminal receptacle and the spermatheca. There's just sperm everywhere. It's impossible to count, right? And this is 24 hours later. Now, if you do the same thing where it's an Americana male who mated with the virilous female, you immediately see that there's just not a lot of sperm in the reproductive tract. You might see one sperm here or there that's kind of swimming in the seminal receptacle. Okay. Now, we've done this over time. We know that these females may successfully transfer, the male successfully transfer sperm. So here's a quantification of that. So we looked 1 hour, 3 hour, 6 hour, 12 hour, 24 hour. And by quantification, I mean broad sort of qualitative categories. This was done blind. And you can see that basically within the conspecific cross, females maintain lots of sperm all the time, whereas in the heterospecific cross, sperm enters storage, but then is lost. So this, I'm just showing you data from the seminal receptacle here. Okay, so there's some sort of sperm ejection phenotype. However, this doesn't really explain why we get 1% success, because by 3 hours, females are starting to lay fertilized eggs. So there's a second layer of something that we're not understanding. This is going to become even clearer when we look at the more closely related species. So this is exactly the same phenotype, same, it's basically just the colors change because it's Nova Mexicana now. And now we want to ask, well, what happens in this particular cross with Americana males and Nova Mexicana females? And at this point, we had a bit more tools that allows us to do quantitative assessment of the number of sperm in storage. So here's again the reproductive tract, and these sperm have tagged GFP sperm heads. So now what we're going to do is look over time in each of those different compartments, the bursa first. So this is where sperm gets transferred. Both male types transfer equal numbers of sperm, about 400. You see that an hour later, but then you don't see any of it because the sperm either was ejected, so females don't store all the sperm, but the amount that they store goes into the spermatheca and the seminal receptacle. So when we look in the spermatheca, they store about 100 sperm, but there's really no difference between the conspecific and the heterospecific male. Okay, so far, okay. Now, in the seminal receptacle, there's a slight difference in the number of sperm that a heterospecific male is able to, or at least that gets stored in the seminal receptacle. That difference is, modest, but it's significant, at least in a couple of time points. And this goes back to what I was saying about the spermatheca being the long-term storage. See, the number here is about 100, and here's the number is quite small. So we think what actually happens is each time there's a fertilization event, sperm moves from the spermatheca into the seminal receptacle. They try to fertilize, some do, some don't. So our data of where the sperm are doesn't really answer why this rate is so low. You see these bars are much higher than this one. Okay, so in addition to the sperm ending up in a incompatible environment, they kind of get lost, but they also, even when they're just not able to fertilize. So I think there's some sinister biochemical issue going on. Okay. So that's all we know about the phenotype. We're doing sort of other experiments to look at the ability of sperm to maintain capacitation or motility in the reproductive tract. But that's all we know, right? So there's obviously some biochemical level of incompatibility that's happening that we need to uncover. So now I'm going to just sort of move to all the genetics and genomics work that we're trying to do to understand what's the cause of this incompatibility. My first efforts were always to do unbiased genetic mapping or positional mapping, QTL mapping, if you will. And we can do this because we can generate hybrids in the reciprocal cross, and then through females, we can recombine those genomes and shuffle them and then basically do standard QTL mapping. So both my colleague, Andrea Swigert, and I have done this, and we both concluded that there are 2 chromosomes that are involved. So these flies have 5 different chromosomes. We map them to 2 of them. One of those chromosomes has a very large effect QTL, but you notice that's a pretty hideous QTL. It's very large because there's an inversion that does not allow us to map with fine resolution in that chromosome. And then there are other smaller effect QTL on this chromosome, which we can see the recombination to. Now, what's important is that both of these QTL account for about 30% of the phenotypic variance. So there's lots of other potentially small effect QTL that we're not mapping. And so this was, this was fine. We needed to move to a different approach to try to get at what possibly could be some of those male candidates. And so the first thing I did after that was to try to actually just profile the gene expression within the male reproductive tract. I reasoned that genes that show expression bias in the male reproductive tissue likely have some male reproductive function. This is, as you'll see later, a very simplistic way to look at it. So we did RNA sequencing on the testes, accessory glands, and the ejaculatory bulb. And I want to point out that the accessory glands produce lots of seminal fluid proteins, somewhere between 200 and 300 proteins, that we know from other work in melanogaster are involved in many of the critical post-mating processes in females, things like inducing egg production, storing sperm, right? Reducing female receptivity to remating, so on and so forth. And some of those phenotypes might be related to the incompatibility that we're seeing. And so this was really just a resource generation exercise back then meaning 2016. We didn't have full genomes, full annotations for all of these genomes, for all these species. And so this was an effort to do that in addition to answering the biological question. So, you get a long list of genes. Testes are kind of, if you don't know this already, they account for about 15% of the gene expression capital in the fly genome. And you can see that here. So out of all of those male reproductive genes, of nearly 85–90% of them are testis biased. This is not unusual. We knew we'll see this. And then here's a small set of quote unquote accessory gland or ejaculatory bulb bias genes. Now, not surprisingly, male reproductive genes in the species evolve fairly quickly, just like in other Drosophilas. So we were not surprised to see this. We, at this stage, wanted to identify those proteins, right? Those that are specific to the accessory glands. We suspect that our candidate genes are amongst those, right? And this list here, so this one is basically those that don't contain a predicted signal peptide. Those are the ones that do contain a predicted signal peptide. Our thinking was that these are the more likely to be in the seminal fluid. That reasoning is fine because there's lots of proteins that actually don't have signal peptides that end up in the seminal fluid. But you can see those that do have a signal peptide tend to have a higher rate of evolution. So this is just looking at DNDS. Usually when I give some talks, I have to explain what this is, but you guys know what DNDS is or omega is, right? Right, so where are those in the genome? Remember, now we did some QTL mapping, so we have some positional information about where candidates might be. And now we have some candidate proteins that we think are in the seminal fluid. Where do they reside on the genome? So here's a sprinkling of those. You can see the red line here is the genome average omega or DNDS ratio across the phylogeny. And I sprinkled on all the SFPs that we identified, the seminal fluid proteins that we identified through that RNA-seq approach. And here's our QTL. So our QTL is on chromosome 2, and then we have a few on chromosome 5. Incidentally, some of those high substitution rate proteins are on chromosome 2 and coincide with our QTL. So this is fun. These are interesting candidates. And so we're sort of functionally trying to characterize what those do. But we also wanted to use other approaches. So we had, at this point, several genomes sequenced from multiple strains. Back when I was doing this work, there's the obsession with islands of speciation and all of that. I think that obsession waned a little bit. But we had data to maybe ask this. So we basically looked at a normalized metric of differentiation. So this is basically divergence between species normalized by polymorphism within species. So here I'm just looking at Americana and Virilis, and the blue dots indicate windows that are above threshold. So this is the top 5th percentile of those values. And you can maybe say that there's a overrepresentation of those blue dots on chromosome 2. Remember, this is an inversion, right? So that is not necessarily a smoking gun, but these differentiation windows could be due to other things. At least they're there. This is now between Americana and virilis. Now, when we look between Americana and Nova Mexicana, remember those are the more closely related species, it's flat, right? Because it's testament to how closely related those two species are. You don't see any of that level of differentiation that you see between the more distantly related species. Okay, so far this is, okay. We didn't really get to something super sharp, but I think this, at least in the lab, we're interested in characterizing many SFPs and multiple Drosophila species. Whether these end up being our star candidates or not is yet to be determined. What we wanted to do, though, is question that RNA-seq approach and use a much more refined approach by using quantitative proteomics. So one of the main attractive things about this system, like I tried to sell earlier, is that you can identify all of the male ejaculate proteins, right? Now, the approach that I just explained is not very direct, right? You're just looking at RNA expression within the male reproductive tract. But males are transferring those proteins to females, and so technically you should be able to identify those if you have a way of differentiating male proteins from female proteins. And this has been done several times. It's been done in melanogaster to identify all the seminal fluid proteins. We used a slightly different approach, which is to do isobaric tagging for really, really fine scale quantitative analysis of those. And so what we did is we take females that are unmated, throw them in liquid nitrogen, right, and freeze them for later proteomic analysis. We then took females, mated them to their conspecific males, and the moment that mating ended, we threw that female in liquid nitrogen and extracted proteins from the reproductive tract as well. Okay, so the idea then is, if there are proteins that are present in the just mated females that are not in the unmated female, that tells us that those proteins had to have come from males, right? So this approach we used on the 3 species that I was talking about. And so this is just a volcano plot. Never mind what all the colors are. Basically, the blue and purple is what we infer to be the transferred male proteins. And I think we get about 200 of those. So this approach is pretty good. It works really well. What we might be lacking are proteins that don't solubilize because of the way we prepared those proteins. But this gave us a pretty solid list of all the SFPs that we think are in the sample. Now, you might be wondering to yourself, okay, so how many of these overlap with your RNA-seq approach? 1/3. That was disappointing to me, but whatever. So this is now our canonical list of transferred SFPs. The ones I showed you in the previous slide, half of those basically remain in our analysis. But this also, you can take a look at this bioRxiv preprint if you like. It should be published soon. But what that really showed us too, that there are nuances to identifying diverging signals between species that we didn't appreciate before. And that is, those SFPs, not only do they evolve rapidly in terms of amino acid sequence, but they also duplicate at absurd rates and they jump around all over the genome. And so this is an angle that we really hadn't appreciated. And it's actually happening in a weird way. This is a plot where I'm showing basically the top half of the circle is one species, the bottom half is another species. Hopefully you can see them. The gray lines are basically the synthetic 1-to-1 orthologs of those one of those SFPs. But the green lines are cases where that SFP is present either in multiple copies in the other species, or has duplicated in tandem, or has duplicated and jumped to a different chromosome. Okay? So I hope you can appreciate that chromosome 2 has an unusual number of SFP duplicates that either jump in tandem on chromosome 2 to other species or are on different chromosomes. Okay, so this is a level of complexity that I was not hoping for. But this is another angle that we're pursuing, is what is really the mode of SFP evolution beyond just sequence divergence? Okay, so stay tuned for that. Okay, so that's really our efforts so far, with some additions that I'm not including about how we identify the male components. Okay, so our QTL mapping was kind of our big hope, right? And obviously you saw that resulted in, identifying, some regions, but really not explaining all the variants. Now, ever since then, when I started the lab, there's this 4th species that I haven't been talking about. We didn't have a genome sequence for, we knew nothing about other than the fact that it existed and it was placed there in the phylogeny. Once we sequenced the genome, what we discovered is that it actually carries no, well, it's got one tiny inversion on the 5th chromosome compared to Americana. Okay, so this was an aha moment, right? Because that means that we can use this species, the species pair, to find map the reproductive incompatibility, at least in both species, right? So we immediately jumped on this. We generated a mapping population. We wanted to confirm that there's free recombination throughout the genome. That's what this mess is supposed to show. And then we wanted to ask, well, can we reconstitute genotypes that have rescuing capability in an advanced intercross? And the answer is yes. So you can see that in these F3s, we're able to get genotypic combinations that are really, really high, meaning that we're able to get phenotypic combinations that rescue. And so that means that it's basically mappable. I was going to show you some of those results. Actually, no, wait, I do have some of them. This is now mapping the female trait. Right? And this is, this is just like 70 individuals from the mapping population. We have about 500. And so, these two peaks here pass significance, but actually these could be artifacts of being either centromeric or near the telomere. But there are, you can see some lower valleys here that might pop up as we increase the sample size. So stay tuned for that. I mean, we're very excited about this because it is going to work, I think. Okay, so now I'm switching really to the female side, because the female side is much more complicated in that, unlike the male protein candidate set, like the way we use proteomics to identify those, can't do something quite similar to females because everything happens within the female reproductive tract. It's basically the full set of proteins in the female reproductive tract are candidates. And when we did that proteomics on the unmated female, we identified about 3,000 proteins or something, right? And so that's about 20% of all genes in the genome. And so our approach differs a little bit. We still looked at some of the sort of gross genomic features. So we did RNA sequencing on the female reproductive tract. I was not yet brave enough to do all the separate different compartments. We're working on that now. But for now, the analysis really just includes RNA sequencing from the lower reproductive tract and then from the ovaries. Here's just a plot of those genes. We get about 120 that are specifically expressed, or I should say, have heavy bias expression in the lower reproductive tract. And so we wanted to ask sort of similar questions. As a category, do those genes evolve rapidly compared to the genome average? What's their rate of evolution compared to the male reproductive proteins? And the answer is that they don't evolve on average as quickly as the male proteins, which is something we kind of suspected, but it was a bit surprising. Now, it's not 100% true, because if you break these reproductive tract proteins down by functional category, namely those that contain signal peptides but are not transmembrane domain proteins, you get a bit of an elevation up to 0.25 DNDS. So this is pairwise DNDS between Americana and Nova Mexicana, the more closely related species. So it gets close to being similar to male reproductive, male SFP-like, but not as a whole, as a category. But I think this is reasonable because we're conflating so many different transcripts from the reproductive tract of females. This is just another way to look at it. We're just looking at amino acid conservation rate, and they're fairly conserved. But like I said, the key message here is that if you break this down by specific subsets of categories, they have a much higher elevation of elevated rate of DNDS. Okay, so it's just a quick aside, because I think I get this question often is, so what kind of interactions happen between male reproductive proteins and female reproductive proteins? And we don't understand it very well. But just looking at the types of functional categories that are enriched in our data set, it's largely things like proteases, hydrolases, these sort of interactions where we think males transfer proteins that get transferred by other female proteins, or by also accompanying male proteins to degrade or to cleave off parts of that protein to make it active, and so on and so forth. And that's why many of the enriched Go terms in these reproductive tissues tend to be peptidases, essentially, right? And so it gives us kind of an expectation that maybe that's kind of the dynamic of interaction between them. So, the female reproductive tract, many things happen in the female reproductive tract. It's kind of it's the business end of things, right? And once an ejaculate is transferred, lots of things happen, not only in the female reproductive tract, but physiologically and behaviorally to the female that are induced by these interactions. So here's just a review that has a nice figure that lists many of the possible things that happen in the female reproductive tract, from inducing all the things that I said earlier about seminal fluid proteins, but also things that are perhaps not obvious, like immune responses or changes in kind of fertilization capacity and so on. And so we sort of wanted to ask whether assaying the post-mating female response can help us answer some questions about what could be happening in the heterospecific cross compared to, say, the conspecific. And maybe that can give us clues as to what molecular mechanisms happen within the female. So it's a really simple experiment. You just do RNA sequencing on females that are unmated, and then you do females that are mated to their conspecifics, and then females that are mated to their heterospecifics. And here we're only looking at we looked at several tissues, but I'm just going to talk about the reproductive tract data. And so once we get all the data back, we can just look very broadly at general expression profiles and how those differ between our treatments. So the shapes indicate the time point. So we had 3 time points in addition to the unmated. And then we have obviously 3 treatments, unmated, conspecifically mated, heterospecifically mated. This is just a multidimensional scaling plot. And you can see that replicates group together. That's good. But importantly, the 2 treatments don't group together in the early time stages, right? So at 3 hours, the conspecific mated females group differently from the heterospecific females. That indicates to us that their expression profiles are fairly different, okay? Now, this difference is reduced as you go later in time. They're still different, but it's not as dramatic as early in time. Okay, so what's going on here? What we tried to do then is basically group genes based on their overall expression profile, just using some clustering approach. And you can see here that this clustering works okay. It never works okay, but this is kind of a best case scenario. If you look at the enriched categories among all of those genes, it's the stuff that we expect. So there's some peptidases, there's some proteasome complex for degradation of nascent proteins, and so on and so forth. So that's fine if you like to look at those lists. What we're mostly interested in, though, is which clusters contain cases where the conspecific and the heterospecific differ in their expression profile. And if you squint long enough, you'll see that there are 2 clusters where there's many genes that are upregulated, so to speak, in the heterospecific cross, but not in the conspecific cross. There's one cluster that has the opposite, but that's really the only case, those 2 genes. Okay, so then we ask, now let me just actually show you the numbers of how these pan out. So they're higher in the heterospecific. There's many of them across the different time points, but very few that are higher in the conspecific. So that's an interesting pattern. There's just rampant upregulation in the heterospecific cross. And if we now take these and ask what is enriched among those, we see something interesting, which is that you get many terms that is about response to bacteria, immune response, and so on. Okay, so what's that about? Well, let me first kind of do a quick aside to explain how immunity works in flies. This has been pretty well worked out. Basically, there are these peptidoglycan receptors that recognize bacteria on the surface of the cell membrane. And a cascade of stuff happens. It activates this master regulator of immune responses called relish. It's a NF-kappa B. And that is a transcription factor that then turns on all the antimicrobial peptides that we're detecting in the data set, right? And so we asked, what is relish doing in the data set? And it definitely is being unusually upregulated early on in the heterospecific cross, not in the conspecific cross. Even though it does get activated a little bit in the conspecific cross, just the extent is much larger in the heterospecific cross. Okay, so that's interesting. And so that then must lead to downstream activation of those effector proteins, these antimicrobial peptides. And that's what we see. Here's just an example of some of those well-known antimicrobial peptides. You can see it's always turning on much higher at 6 hours in the heterospecific cross, but not the conspecific one. Now, there's a general stress response that we don't understand as well. that is happening. So in general, females are responding to these heterospecific males as if they were some kind of infection, there's some stress induction, right? So that's very peculiar. And I think at the time, my thinking was that this is a response to seminal fluid proteins, right? I still kind of think that, but I'm not showing you this data. We wanted to know whether microbes could be playing a role. We actually knew nothing about reproductive microbes. And so we've done some experiments to show that, and this is actually published too, that yes, having microbes induces an immune response, but removing microbes by making flies azenic or germ-free, you still get some sort of immune response. So that indicates to us that immunogenic responses in females can be mediated by reproductive proteins. And furthermore, they can be mediated even strongly, much more strongly by divergent reproductive proteins. And that's really the hypothesis that we're interested in exploring. There's a couple of papers and precedents for this. So Ted Murrow and Paolo Innocenti published a couple of papers a while back. One is a review that says, okay, here's how you would actually confirm that this is an immunogenic response by reproductive proteins rather than bacteria transfer, which was at the time not really even a thing. So, okay, the big question is, does this explain the reduction in fertilization? That's the 1 million pound question that we don't know the answer to yet, but that's what we're working on. Okay, so our main interest now in exploring the female reproductive response is to really do proper inference of the cascade of gene expression changes in the female after mating. Okay, so what I showed you was 3 time points, unmated sample, what I'm calling sparse sampling of post-mating time points. What we've been doing lately is to do much more dense time sampling with the idea of being able to actually model the gene regulatory network itself. Okay, so the idea that is if you know at a very dense time scale when genes change in expression, you might be able to build lead-lag relationships between protein activators and suppressors and so on. At least that's the idea. So we've done a pilot experiment to explore just that. We've sampled reproductive tissues every 15 minutes for 6 hours. And then we lost interest and we started doing it for every half an hour for another 6 hours. This is the work of grad student Gagny, who's very courageous to undergo this. And this data is this is a very large data set, very messy. 2 things that I'm going to kind of share with you now is that you don't need to sample that densely, basically, right? The gene expression changes are not there. There's no bursts, at least not that we can detect, that are affecting immediate changes that are super short term. There seems to be 3 modalities of gene expression change. So genes are either from early time points steadily going down, or there are ones that are steadily going up. But in the system, and then there are ones that are just going up kind of in the middle. So these are highlighted here. So these are the things that go up late. Here are the things that go down, and some stuff here that's sort of kind of in the middle. Now, some things that we didn't appreciate would happen that, when you do this level of dense time sampling, your power to detect differences is ridiculous. So what that means is like half the, basically like every gene is significant, right? Every gene changes at some time point. And so parsing through how to basically build those lead-lag relationships becomes non-trivial. And actually we were having some success in at least identifying some things. What turns out to be an issue is that, you need some priors about which genes are involved in which networks or which processes to get a better handle of those GRNs, right? If you have none of that information, you get hundreds and hundreds of networks that are plausible, equally plausible, right? So we're basically building a catalog of priors, mainly by orthology to melanogaster, and we're using that to build those GRNs. But overall, this approach works okay. Some colleagues asked me, well, why do every 15 minutes? And my answer now is, so you don't have to. Okay. All right. So that's all we know so far about the female reproductive tract. I'm running a bit over time, so I might breeze a little bit faster through this last project. this is really just a work in progress. I wanted to tell you guys about it. It's the first time I'm actually talking about it. And so this is a case where we can create sterile hybrids in this cross between Nova Mexicana males and Varroa females. And like I said in the beginning, you would think that this would be a rampant phenomenon, and it probably is. I think all the classic cases of hybrid sterility that have been studied, both in Drosophila and elsewhere, rarely involve the Y chromosome. There is a case actually by my colleague Andrea Swigert where it's Americana and Varroa hybrids, but it's later generation hybrids, I think F2s or F3s, where you can then get basically uncover autosomal Y incompatibilities, right? And obviously because now you're reconstituting recessive, homozygous recessive individuals. This is a case of F1 hybrid sterility. So let me explain how we recover that. So if you make a hybrid between Varroa females, Nova Mexicana males, that male is sterile, okay? We can rescue this sterility by essentially introducing the Americana Y chromosome in place of the Nova Mexicana Y chromosome. We can do this very simply, like I'm showing here, by just basically backcrossing males to Nova Mexicana females until you get an Americana Y and an otherwise Nova Mexicana genetic background. And if now we make a hybrid with Varroa, we get this individual, and you'll notice that the only difference between them is this tiny Y chromosome. And that individual is completely fertile. I mean, they're absolutely fertile, okay? And here's some of that data. This is a complicated plot. I won't go into all of it. The basic idea is here's the Varroa Nova hybrid, totally sterile. If we put in a Y chromosome from Americana, totally fertile. And now we can do the reverse, right? We can create a hybrid between Americana and Varroa, which is typically fertile. But now we introduce the Y chromosome from Nova Mexicana into this Americana male before making the hybrid, and also completely sterile. Okay? So what that means is that the Nova Mexicana Y is necessary and sufficient to cause sterility in a Varroa X background, regardless of autosomal genotype. Now, there's only been one paper on this, or actually I should say two. One of those papers was by Heikkinen and Lumi, the Finns who really identified the detailed genetics here, and one by my former advisor, Alan Orr. But since then, there's just really been no work on it. So I picked this problem up thinking that, we can just throw genomics at it and that'll solve everything. And then my reasoning was, well, the Y chromosome is a small chromosome. There's only a handful of genes. If we just know what all those genes are, we'll solve it, right? At least that was my naive attitude. Okay, so what is the sterility itself? So it's a defect in sperm individualization. So all you need to know really is that in Drosophila, spermatogenesis happens within a sperm cyst, and then that cyst, it's all the meiotic and mitotic divisions happens within that. You end up with, I'll say 64 for melanogaster, but you'll notice that number is weirdly different for Varroa, right? And once those divisions take place, and then the elongation of the sperm happens, those sperm become individualized through this process that I'm showing here, this caspase-dependent separation of that cytoplasm between each sperm bundle, or each individual sperm, sorry. Okay, and we think that this process is kind of wonky in the hybrids, okay? So here's an image of our, this is just a wild type. Basically, at the end of the testis where this weird loop happens, this is the seminal vesicle where basically individualized motile sperm exist. You'll see there's almost a very clear barrier here between now, these are still in the sperm bundle. What I'm tagging here is just the sperm head. So you're only seeing the head fluorescing green, okay? And the rest of them are sort of in a coil that you can see. But once they pass this barrier, something magical happens, they just unravel and swim away, okay? So if you cut here, you cut this part, all the sperm that's in here will leak out, and you'll see this is really spectacular. It's like looking at a light show. They're all just kind of swimming in circles, right? It's sort of exactly like this. If you do this in the sterile hybrid, you don't see that. Only this junk here comes out. You see a lot of disordered sperm heads and so on. All right. Now, we thought we might just be able to do some EM sections near that area and be able to just see defects in the cyst bundles and solve it there, right? Turns out it's not that simple. So there's basically no difference. If you look at these EM, let me just show all of them. If you look at these EMs, and we've looked at lots of these images, there's just no difference in gross morphology of the cis bundles, no difference in the sperm heads. None of the slices that we had showed any particular defect. I'm not going to go into this. This is just demonstrating the same thing I explained before, but now keeping it in its kind of weird coiled shape. And again, if you open that receptacle, the seminal vesicle, in the sterile hybrids, you basically get very few, if any, coherent looking sperm. Okay, so that's the phenotype. Oh, and I should say, so if you guys know, in melanogaster, there's 64 sperm per cis bundle. We get 36. So if you can work out how that can happen, please let me know. It doesn't make sense, but anyways, we've counted hundreds of these, and they can vary sometimes. It could be 36 or 40, right? Our actual distribution is like 36 or 40, but 36 is much more common. And this is in both fertile or sterile, it doesn't matter. So I don't know, that math just doesn't work. If somebody figures it out, let me know. Okay, we'll walk backwards from 36, divide by 2, divide by 2, divide by 2. You get to 9 pretty quickly. And I was like, well, how do we get there? Anyways, so why I really thought this project was tractable was that, we were, at the time I was a postdoc, we were sequencing lots and lots of genomes, and one of my jobs was to, find Y contigs, Y chromosome derived contigs in those genomes. And this was fairly straightforward to do. If you have male and female short read data, you basically map to a male genome, and all the male reads that basically don't match, basically sort of a matching thing, right? Like the coverage of Y contigs should be a lot lower when you have the female data. Okay, so that's how we infer that. And we can do that with pretty good confidence. Here's just the data from Nova Mexicana and Virilis. You see that we infer Y contigs as being those that are unmatched by female reads, right? And so it's very easy to get a clump of those. Some not so easy to identify, but we get a good set of candidates. And my thinking at the time was, okay, well, we just assembled those Y contigs, find all the genes on them, and just compare the species, right? Yeah, wrong. It's really not that trivial. We don't see any smoking gun differences. There are cases, ambiguous cases of maybe this gene is there, maybe it's not there. Very often those end up being transposase coding sequences, right? And I don't need to tell you the Y chromosome is just littered with what we used to call junk DNA and junk stuff, but it's potentially playing some important role. Okay, so the last thing I'll tell you about is we then thought, well, let's ask what is happening within the testes at the expression level. And this is a very simple experiment. We take testes out from fertile hybrids, sterile hybrids. Remember, they only differ by the presence of by the Y chromosome, right? The steriles have Nova Mexicana Y, the fertiles have Americana Y in an otherwise hybrid Virilis Nova Mexicana background. Okay, so when we do RNA sequencing and we look at the MDS plots, here's the parentals. They're in different planets, as we may expect. Here are the hybrids. They're very close to each other, so they're very, very, very similar. There's a slight difference between them. And so when we compare them directly, there's not a lot of genes that are different between them, right? And no functional categories enriched here. Only one of those genes is on the Y chromosome. It's this one here, right? And so interesting pattern. We thought, well, you know what? The Y chromosome is expressed in a very small subset of the testes and mainly in the spermatocytes. So maybe our resolution to detect that difference is just not there. And so I thought, okay, well, this was at the time when everybody was doing single cell sequencing. So we thought, well, let's just do that. And actually, this is fairly new data. I'm still analyzing it this week. So just to quickly walk you through it, we did the two hybrids, the fertile and the sterile hybrid, and one of the parentals. And actually, this is just a mishap. We lost the Nova Mexicana sample in the prep. It basically didn't work out really well. This was a pilot, okay? So don't throw too much into it. We were able to annotate those cell types more or less okay. So this lineage here, you can see basically the sample distribution. This lineage here is the hybrids, and here's the Virilis sample. And these are the germ cell lineages, the ones that we are interested in. Now, I hope you can notice that at the late spermatocyte stage, the fertile hybrid and the sterile hybrid are actually very, very different, right? And so we asked, what is the difference between those two clusters? Again, there's only a handful of genes. And guess what? 70% of those are the same that are in the previous plot. Okay, so all of those differences, ouch, okay, here we go. All of those differences that we detect in the bulk RNA sequencing overlaps with what you see at the difference between the two late spermatocyte stages. Okay. Okay, what could that mean? We don't know yet. Here's just sort of a view of that. So what we've noticed is that many of the genes that are different between those two tend to be unique to one sample. So you can see cases here where it's up in the fertile hybrid. It's not even expressed in the parental Virilis sample, right? And vice versa. So there are cases where it's expressed only in the late spermatocyte in the sterile hybrid, but not expressed in the parental. Maybe it's expressed in Nova Mexicana, we don't know. So we need to really get the end of that data. Okay, I'll stop here. I went way over. Okay, I just told you that Virilis is a great system for working on this stuff. We think the gametic incompatibility is complex in males, potentially simpler in females, but we're yet to get final mapping data on that. I showed you that heterospecific mating induces a weird post-mating response, this immune response, and we're not sure if that's involved with the incompatibility, but we're looking after that. And then finally, I told you that the sterile and fertile hybrids have really subtle differences in transcript abundance, but we're sort of, we're really throwing everything at this. So we're doing like small RNA sequencing, right? Just to give us any clues. Just the gross morphology of the Y could also be involved, and that's something I didn't talk about. Okay, I'll stop there. I highlighted some of the work of Amisha, grad student in the lab. Martin, who's a former postdoc, did the proteomics. Yagnit is doing the GRN inference with the RNA-seq. And Anne is the heroic person who did all the single cell sequencing. And a lot of this work was really started in my postdoc mentor's lab, Andy, in collaboration with Mariana. This is our website, thanks to the funding. We're always looking for postdocs if you're interested, so reach out to me if you're like this work. Happy to take any questions if there's time.
Zoom folks, you could raise your hand or say it in chat, and folks in the room, questions? Oh, I'm going to make it so we can see
Trying to
just
get
a moment here.
so I can see you better.
Aha, I see a question. Wait.
Oh. That was a great talk. I want to study something that hangs out by Saki also. I was curious about when you mapped the sterility. Oh no, I don't like this. You mapped the sterility to the Y chromosome. You did Americana and Nova Americana crosses. Is that right? And I thought that those crosses didn't work based on the previous results with male Americanas. Yeah,
no, good question. So in the reciprocal cross, we get about 20% fertilization success. So if you're not using Americana males, you still get some, right? You still get like 1% fertilization success. So you just have to like use up 2 shells in the incubator with vials, right? You'll still get a handful if you do that cross. So it's not 0% fertilization success. But yeah, we typically just use the reciprocal cross to do a lot of the hybrid generation. But yeah, if we want to get, so Americana Nova, it's like 2%. Americana Virilis, 1%. But that 2%, if you put like 1,000 flies together, you'll get a handful.
Got it. Thank you.
Good intuition.
Yeah, I have a question about the differences between the fly species. Are there differences in morphology of, say, the sperm or the different reproductive tracts?
That's a great question. So the sperm, we don't know. There are slight differences in length between the species. So there's a paper by Scott Pitnick from, I think, 99, where he looked at sperm length in many species, including the Virilis group. There are slight differences in sperm length and also in the female seminal receptacle length. As far as sort of reproductive tract morphology, internal morphology, we haven't looked, but to the naked eye, doesn't look like it. We are actually interested in looking at genital morphology, but actually relating to the immune response. Many of my colleagues suggested, and I think it's totally plausible, that the immune response is actually a secondary effect of wounding, right, due to some kind of divergence of genital morphology. So, but we don't know yet. We were talking to a colleague to help us kind of look at those. Not trivial to do that. But beyond that, yeah, so sperm length is kind of the big one, right? If you have incompatible seminal receptacle and sperm length, that might reduce fertilization success. But the length differences to me are just not that big. So, if it's causal, I'd retire tomorrow, right? But it's probably not long, not the differences are not big enough to lead to that, I don't think. But that's something we're still interested in exploring. Yeah, thanks for that.
Hi. So Virilis is widely distributed across the world. Can you see any sort of incompatibilities within species, like between populations that are far apart?
Yeah, we haven't looked at Virilis populations. So, this sort of worldwide expansion is actually, it's known exactly when it happened. It happened in the early 1900s. So I presume that's just not long enough for that to be like a meaningful divergence time. However, we did look at different Americana populations. So in my 2012 paper, we looked at gametic incompatibility between different populations. And even though it never reaches such low levels, it does definitely significantly vary. So if you have a isofemale line, say from the east side of the US, and take a female from that, mate her to a male from the west, you might see a reduction in fertilization by like 5% or so, right? Which is significant in our case, but nothing as drastic as the between species, which is again a testament to that, yeah, these probably are evolving pretty rapidly, right? Now, our Nova Mexicana, it's a bit of a sad story. We only have like 4 strains, and we don't have a lot of genetic diversity sampled from those guys. And we think that population is just low NE, so low segregating polymorphism. Lumii, we just don't have that many strains for to do the same thing. But for Virilis, we just haven't looked. We do have quite a few strains, so we probably can. But yeah, I would say yes, there's a reduction, but not as dramatic as between species.
Wait, can I ask my question? Do you think that the immune response stuff is due to like male-female conflict things, or do you think it's maybe, the coevolution with pathogens are making the immune stuff shift and therefore,
Yeah. That's a great question. I mean, I've had all the opinions over the last 3 years about this, right? I at first thought, it has to be divergent male molecules. And then I was convinced, it had to be transferred microbes.
Well, I didn't mean the transferred microbes. I meant just, other pathogens causing the immune system to do stuff. And then as a result,
So, I mean, that has been the historical explanation for an immune response during mating. It's sort of the natural selection explanation, right? Which is that during mating interactions, you're exposing yourselves to environmental microbes. And so therefore, the adaptationist thing to do is to launch an immune response as a way to prevent STDs, if you will, right? Or other environmental pathogens. So that's always been kind of, at least that's what I was told is the canonical explanation for that immune response. I think it's still possible that is why that response evolved. What makes it weird in our case is that it matters what the genotype of the male is, which then that means that there's sort of some potentially a second level of activation of that immune response that is not strictly the natural selection explanation. But I keep telling the story often to my friends, which is like, I tried really hard to not work on immunity my entire career, and I've just failed. So like now we have to, why is immunity involved with reproduction? This is kind of the big question. I mean, Brian Lazzaro's lab does a lot of work in this area at Cornell. And so what we were wondering is, and where my interest lies is if this is a speciation phenotype, then I'm interested. But if it's just some fluke of, we have strains that have this bacteria and strains that don't have that bacteria, it kind of deviates away from what the original purpose was. But I'm obviously very curious as to what's causing it. But depending on the answer, my enthusiasm might switch. Yeah, thanks.
Great talk. I have another question about the immunity part.
Okay,
Have
go
you
ahead.
thought about like decoupling the effects of like sperm, or is it maybe seminal fluid alone?
Yeah, yeah.
Or if it's
Yeah,
just like the mating and not actually transferring anything?
Yeah, so if this was melanogaster, we'd be able to do that experiment because we would have strains that either produce only sperm, only seminal fluid, or none, right? We don't have similar resources in this species. So, I mean, one thing I guess one could try is, what we've done before is we've done consecutive matings between the two different species. So, because these guys will remate pretty readily within a few hours. The one way we did this experiment was we separated them like a day apart, right? So we'd do a mating today and then do a mating the next day. But we didn't look at immunity, right? We looked at just reproductive success, which it did. Well, the hope was that, if you have the conspecific ejaculate in the presence of heterospecific sperm, do you rescue fertilization? The answer was no, but it could be also that it's a day apart. And I was asked this recently. So if you do the same experiment, but now don't space them that far apart, do you, first of all, get the rescue of heterospecific insemination? That's one question. And then the other one would kind of get at your question. But the paper by Innocenti and Morrow, actually, that's how they discovered the heightened immune responses. They did consecutive matings, and it was after the second mating where the immune response was really heightened, right? So long way to say no to your answer, to your question, basically.
Thank you so much.
Yeah.
Any other questions? Okay, let's thank Yasir again. Yeah, thanks. That was great.
Thank you so much. Yeah, that was a lot of fun. See you.